big now, but I want to share with you some of very recent experiments we've been doing focused on the effects of oxytocin on the salience system in the brain and its interactions with other networks. Just to remind everybody, a lot of people I think in the room probably know a lot about oxytocin, but oxytocin's been around a long time in terms of research. It was originally discovered by Sir Henry Dale in the UK, and of course it's most well known for its smooth muscle effects in promoting uterine contractions during birth and also milk ejection. But with work starting by us and by other groups in the UK and the US, particularly in the mid-1980s and 1990s, we discovered that it's also released within the brain itself, particularly high concentrations in the cerebrospinal fluid. And there's also a projection system within the brain to a number of key areas involved in social behavior and reward. The main findings, if you summarize a huge amount of work, really in animals are shown here. I'm not sure whether this one is going to... Oh, there it is. Mostly we found in sheep and also sucata and Tom Insel, particularly with voles, that it's important for stimulating the formation of both maternal offspring bonds and also partner bonds. But also it's very important in animals for social recognition and other aspects related, protection of offspring, mate guarding, and cooperative behavior. And if you like, you can summarize all of these as attention being switched away from self towards external social cues. And indeed, recent work on voles has also started to focus particularly on the effects of oxytocin in relation to the pair bonding on the actual social salience network, rather than the initial focus, which was and still is to some extent on the reward network. Moving to humans, I won't go through some of the controversies, we can discuss it afterwards, but we now are pretty much certain that oxytocin, given by nasal spray, which is the usual way that we give it in order to influence brain function in humans, does get into the brain. It also gets into the peripheral vasculature, and the exact interaction between peripheral and central actions still remains to be determined. But there are a wide range of effects. It's obviously released during sex, birth, suckling, tactile stimulation, social interactions. And as you can see from all of these behaviors, again, they tend to be pushing individuals to be a little bit more other-orientated. Whoops. And of course, the bottom line with the whole field is that all of these kinds of functions that oxytocin appears to be playing a role in could be very relevant for therapeutic potential in a wide range of disorders, the main one of which is focused on is autism. This just shows you another controversy is how long it takes oxytocin to get into the brain. Experiments typically start about 45 minutes after the nasal spray, and this just shows you from the Palaeolis group regional cerebral blood flow changes, which do indeed occur maximally in most of the brain regions containing oxytocin receptors at around 39 to 51 minutes. There are a number of explanations about the critical role of oxytocin, and I don't want to debate which one is necessarily the best. They all have pros and cons, but one of the most discussed ones is the social salience hypothesis, originally from Jennifer Bartz, and it states that oxytocin functions primarily to increase the salience of social stimuli, but, very, very important, but not generally. It is enhancing it in a person and context specific manner. That means that whether it has effects or not, will be modified by traits, psychiatric traits, personality traits, gender, sex, and also your current state of motivation. I prefer to sort of regard oxytocin generally as amplifying whatever your current social priority may be, and of course that can vary quite markedly. This just gives you an example of early processing in the attentional system of emotional faces. This is a masking paradigm where actually subjects only see emotional faces for 20 milliseconds, but that has effects on the salience system and amygdala, and oxytocin enhances that effect, notably for fear faces. But you'll notice for the anterior cingulate here and the insular part of the salience network, the effects of oxytocin in males and females are completely in the opposite direction. What I'm going to focus on next is the question as to whether oxytocin is playing a role in attention switching from internal to external cues. And the insular is particularly important in this respect, being the recipient of interoceptive information and also awareness. And it's possible that oxytocin might be increasing interoceptive awareness. This is from Lucina Udin, showing the salience network is proposed to be involved, very important, in dynamic switching between other networks, such as the default and the central executive network. In this task that we set up for oxytocin effects, we firstly looked at whether oxytocin has an effect on interoceptive awareness per se, that is being aware of your heartbeat, and you tap your finger when you think your heart is beating. And the control motor task is just tapping your finger every time you see a white dot. We looked at the effects of oxytocin on that. And then secondly, we looked at the same task, tapping for your heartbeat, while you have external social stimuli exposed to you, which are emotional faces. In the first part of the study, just interoceptive awareness, oxytocin actually has no effect. Yes, we get beautiful activation of the anterior insular, cingulate, and posterior insular, but there's no effect of oxytocin on that or on accuracy of heartbeat detection. Contrast that, though, however, when you show emotional faces and interoceptive accuracy is significantly decreased. Although right across all of the emotions, in fact, although you see, as you would expect, that negative emotions are associated with an enhanced accuracy. This is fairly well established by others. And if we look at the brain, now oxytocin enhances the right insular response. And it's also correlated with interoceptive accuracy scores, whereas in the absence of the external social stimuli, there's no such correlation. Importantly, also, there's a correlation in the functional connection between the anterior and the posterior insula, with exactly the same correlation with interoceptive accuracy scores. Originally, this is from the Craig review, the idea is that primary interoceptive information is processed by the posterior insula, and the insula receives inputs from many different regions, but you become aware of it when it reaches the anterior insula. In this review, it's considered as a one-way traffic. But what we're proposing is, in fact, that what oxytocin is doing is increasing control from the anterior to the posterior insula, so that it actually controls the impact of interoceptive cues, so you switch instead to focus on external social ones. Quattrocchi and Friston, in a review that very few people actually cite, also predicted that oxytocin may be having effects on prediction error signaling, which will be entirely consistent with these results that we found with the insula. And just recently, there's been a focus on social synchrony effects of oxytocin in both humans and monkeys. You're more synchronized when you have oxytocin. And this paper just came out a few weeks ago showing that local infusions of oxytocin directly into the anterior cingulate increase social synchrony in monkeys. In fact, also vasopressin has the same effect. This is also potentially prediction error type signaling. In the last part of the talk, I'm going to focus now on looking at oxytocin effects on functional connectivity, but directed functional connectivity in the salience and related networks in 200 subjects, both males and females, and using a Bayesian connectivity change point model, which I don't have time to go into, but it's based on temporal sequencing analysis, the resting state, and allows you to infer directionality. And when we do this with 19 different regions, we get a unique pattern in the control group of 14 directed connections, and you'll notice that all of these within the different regions tend to be within the regions themselves, not between regions, except for the ventral tegmental area projection to the amygdala. But contrast that with oxytocin, the pattern, you get this pattern in 80%, right? The subject is really very robust. You get clearly a big change where suddenly now the directed connections are between regions, between the reward regions and the salience network and the amygdala. And we even get sex-dependent patterns. These only occur in males. Similar pattern, though, that it's between regions, not within them, that oxytocin is working. This shows it more simply, and you can see not only does oxytocin promote connections within the salience network, but also between the reward network and the salience network, and between the reward network and the amygdala. The oxytocin effect in males additionally involves amygdala to salience network connectivity direction from sort of bottom up to the anterior cingulate and also from the ventral striatum. I just wanted to say it sounds like a fairly exotic method we've used here, but you can demonstrate exactly the same effect of oxytocin on increased interregional connectivity using, in this case, independent component analysis. And one of the members of our group, Xin Fei, will be giving a talk on Thursday morning on this. So I just wanted to show that you can see this effect of oxytocin using different approaches. So in summary, I'd like to share with you that oxytocin influences particularly the salience network, particularly by promoting its interactions with other networks involved in processing social signals. It enhances interest in person and a context-specific way towards socially salient cues, and importantly, particularly in the context of maybe in autism, it has some important sex-dependent effects. Oxytocin may also particularly function to influence prediction error signaling, to control integration of interoceptive and exteroceptive cues, and to promote social synchrony. And the concept of oxytocin turning up the gain was in a recent article as a result of a trainer's group article on rats showing increased activation in the brain of animals looking at social situations, and of course from the kind of work that I've been describing. It just remains finally to thank my members of my scan lab. In the context of this talk, obviously particularly Ben, Kevin here, who did the insular work, and where is he, Shijiang, who did the connectivity work. And yeah, you might think we have to use the oxytocin for our more social context, but I believe you me, we don't. Thank you very much.